Okay. Yes. So let me just share my screen and we can begin. Okay. So in case there is any lag, please let me know because I do feel that the net might be a little problematic. I'm meeting from a different location today. Yeah. I think I've not altered this. Yes. If you are one of the people who have been attending these extra sessions by me, you will know how I generally go about the session. So, first, what we'll be doing is we'll go through the questions that were posed pertaining to week 6 in the previous year because current year's assignment is still on. So, we'll not particularly discuss those questions but we'll discuss the previous year questions. Some of the concepts might help you solve this year's questions, and also it will be another 10 questions that you get, which will help you, study for your exams if you are appearing for them. So that way also it might be helpful. What we'll do is we'll go through the questions and we'll also understand the associated concept behind that answer wherever we arrive, right? So, yeah, we'll begin with this. And in the second part, we'll finish off the sensory system talk that we couldn't complete in the previous session. And I think because that is also a substantial, that will take a substantial amount of time. So what we'll be doing is we'll finish with the sensory systems today and learning and memory, that bit I will take on as the extra session in the next coming Monday's class, right? So yeah, correct. Let us begin with the previous year questions. So the first one was, what is reticular activating system or RAS primarily responsible for, right? So in this week's lectures, you might have like sir actually began discussion with this particular system, reticular activating system, and there were a few lectures that were dedicated to it. So over here, the simple question that is being asked is, what is it primarily responsible for? What is its primary function? So, the options that you have are, is it specific muscle movement or hormone production or cortical arousal and consciousness or visual perception, right? So, if you have gone through those lectures, it will be easy for you to come to the answer, right, which is cortical arousal and consciousness. So, reticular activating system is responsible for us being in that conscious, alert state, right? There were also discussions in which it was, discussed that lesions to this area also render a person in maybe a comatose state or a brain death state or as well, right? So we'll discuss a little bit about the reticular activating system. So this has been taken up from the previous discussion that I had in the extra session. Wherein I had discussed about particular brain areas and their functions. So I'll just recap that over here because it is uh, of importance over here, right? So reticular activating system, you can see this part is the reticular activate. This is the reticular formation. It is going along the brainstem, and there its processes basically they lead till the cerebral cortex as well, right? So the ascending portion of this reticular formation is basically known as the reticular activating system. What it consists of is sensory axons, which project to the cerebral cortex, as we can see over here, both directly as well as through thalamus, right? So through here also it can go, through the thalamic course, or it can directly also go towards the cerebral cortex. Also, the most important function of RAS, which is what the question is based on, is consciousness, which is a state of wakefulness in which the individual should be fully alert, aware, and oriented, right? Also, visual and auditory stimuli and mental activities can stimulate this system, that is RAS, to help maintain consciousness, right? So, all this we had discussed in the previous session as well. What is another function of RAS other than consciousness itself? It is to help maintain attention and alertness. So altogether, you can see that it is responsible for this alertness, attention, consciousness, these aspects of our functioning. Right. It also prevents sensory overload, that is excessive visual or auditory stimulation, by filtering out insignificant information so that it doesn't reach consciousness. So I had given you an example that it is like whenever you are in a crowded environment, for instance, and you are speaking to a certain person, right? You are focusing your attention to what that person is speaking, right? Although there are so many other things that are going on around you, there might be other conversations that are going on in your vicinity that you can hear, of course, but you are not listening to them, right? So how do we arrive at the difference that it is getting, because that is not relevant to where your brain wants to be alert or attentive to. Your brain, and particularly this system, reticular activating system, what it does is it filters out that extra noise, right? So that although your that stimulus might be reaching your ear, but once it is passing RAS is a place where it is determining that, oh, this is not worth my attention, so it will filter out those signals, right? So that is how you can carry forward a conversation even in a crowded environment. Then inactivation of this RAS, what it does is it reduces sleep or a state of partial consciousness from which an individual can be aroused, right? If the RAS is activated again. But if it is damaged, right? For instance, we talked about a lesion in this particular region, RAS. Then what happens is it results in coma or a state of unconsciousness from which an individual cannot really be aroused, right? It is very difficult to reverse that situation of comatose. The descending portion of RAS has connections to the cerebellum. As you can see, this was the ascending portion. What was the ascending portion doing? It was, throwing its connections till the cerebral cortex, be it directly or indirectly through thalamus. Now, the descending portion is this one, right? It has connections to the cerebellum, as you can see over here, and the spinal cord as well. And it helps regulate the muscle tone, slight degree of involuntary contraction in normal resting skeletal muscles, right? So, although muscle movement is a voluntary movement, right? But despite that, the muscle is always in tone, right? It is not like flaccid and just loose, right? That is the case only in some kind of injury. So if you've had any kind of muscle injuries, you will know that the muscle becomes loose and that tone is lost, right? But that is not always the case. And that muscle tone is also maintained by RAS, right? This portion of RAS also assists in the regulation of heart rate, blood pressure, and respiratory state, right? So, kind of the functions of cerebellum and medulla is what are being described over here, right? So that was our discussion about RAS, or the reticular activating system. Now we move on to the second question, which is what did Eugene Aserinsky observe about eye movements during sleep, right? So was it that eyes were completely still during sleep, or that eyes made rapid jerky movements during certain sleep periods, or that eyes only blinked occasionally, or that eyes moved slowly as well as consistently, right? So, even if although you all might have gone through sir's lectures, but even if you wouldn't have gone through sir's lectures, now as a community, we are much more aware about sleep and stages of sleep. You might be able to answer this anyway, right? So you know that there are multiple phases of sleep. There's a REM cycle, there's a non-REM cycle, even your gadgets, your those smart rings, smart watches, etc. They monitor all these things, right? So, uh, what is actually the case that is going on? It is that eyes are making rapid, jerky movements during certain sleep periods, right? It is not consistent. It fluctuates. It does not just blink occasionally, right? As was initially thought, and they are not completely still during sleep also, right? They continually move, especially in the REM phase, which is the rapid eye movement phase, right? So this was the original paper by Aserinsky and his guide Nathaniel Kleitman. The paper was titled Regularly Occurring Periods of Eye Motility and Concomitant Phenomena During Sleep, right? So it was published on September 4th in 1953 in the journal Science. And this is a snapshot from the actual article. And I have highlighted this portion wherein in this report they dealt with different type of eye movement that was already persistent and already known during that time, right? What was this eye movement? It was rapid, jerky, binocularly symmetrical eye movement, right? So what it means is that in both eyes it is happening at the same time, right? That is what is meant by binocularly symmetrical. So, this is another original graph from that particular paper, wherein what they have, the figure legend that they have written says sample record exhibiting rapid movements in a sleeping subject. RV over here represents vertical leads on the right eye, RH is the horizontal leads on the right eye, and RF is the right frontal. So these are different channels from which recording had been done in that particular subject, right? So, yeah. So, what neurotransmitter was speculated to be involved in the reward circuit, right? Was it dopamine or serotonin or adrenaline or melatonin, right? So, again, in the previous discussion, in the previous class, I had this table in which neurotransmitters were listed and their functions were also listed along with them, right? So, even if you had gone through that, you would know this. And of course, this week's lectures also mention which of these is was speculated to be involved in the reward circuit. And now we know that it is actually involved in the reward circuit. It is actually dopamine, right? So dopamine is one of the neurotransmitters. Uh, which again is involved in the brain reward circuit. So over here you can see the blue path represents the dopamine path, right? So the functions are reward or motivation, pleasure and euphoria. Motor function also we already knew that it is involved in because the loss of dopaminergic neurons or dopamine producing neurons are the ones that actually cause, diseases like Parkinson's, wherein your neuromotor coordination is lost, right? So that also we have studied before. Also, it is responsible for compulsion and perseverance, right? Because if you're motivated for something, you will persevere to achieve that, right? So, this is the reward circuit. Also, in this circuit, there's serotonin that plays a role, although it is more commonly known as a happy hormone, right? Its functions are to stimulate the mood, the memory processing, sleep, as well as cognition, more than motivation per se, right? Serotonin also we have discussed over here. Apart from this is adrenaline. Now, adrenaline is your, response hormone or response neurotransmitter. And melatonin is a neurotransmitter which is more associated with the sleep induction, right? So what happens, and With sleep, it will also balance the circadian cycle, right? Then question number 4 is what happened when researchers created lesions in the lateral hypothalamus, right? Did the animals become obese or the animals stopped eating altogether or the animals ate more slowly or they became more active? So what happened out of these when lesions were created in the lateral hypothalamus, right? So, in the hypothalamus, we studied that there were two kinds of centers that were almost adjacent to each other. One was a feeding center, and one was the satiety center, right? So, over here, uh, the lateral hypothalamus might have been the feeding center, right? Because when lesions were created in the lateral hypothalamus, as a result of that, what happened? The animals actually stopped eating, right? Or, yeah, okay. And this is a representation of the maintenance of the energy balance that normally takes place, right? So normally what happens is, let me just zoom into this. Normally, what happens is that the hypothalamus that is present, it gets a signal from the blood. So this is one of the organs wherein blood-brain barrier is a little loosely present. It is almost negligible in case of thalamus, which has to maintain, the homeostasis. So over here also homeostatically, it gets some signal from the blood, which in turn are getting signals from the leptin and the leptin cycle and the pancreas, right? And they, transfer this signal back to other regions of the brain wherein it is processed and again back to the gastrointestinal tract also wherein ghrelin is present, which is the hunger inducing hormone, right? So if this ghrelin is actually present, it will go to the blood. It will tell them that, okay, I need food, right? But on the other hand, what happens is when this ghrelin is absent or when more leptin is present instead of this ghrelin, what happens? Leptin induces satiety. So it will go and it will signal to hypothalamus saying that, okay, I don't need food anymore, right? So leptin action in hypothalamus goes a little bit like this. We are not going into the details of this as yet, right? So what happens in case of lesion of lateral hypothalamus, there is alteration of various pathways which include or synergic system, right? There are ascending projections wherein there are behavioral changes which lead to reduced food intake, and in the descending projections as well, there is increase in the sympathetic activity, means the you become more prepared or more alert, right? So when the sympathetic system is higher, what happens? You feel less hungry, less thirsty, because your body is prepared to burst out, fight, or something like that, right? So there is again reduction of food intake over here. Question number 5 is what did Ernst Sharrod discover about the nerve cells in the hypothalamus of teleost fish? Right? Was it that they were exactly like computer circuits, or that they resembled glandular cells, or that they produced no secretions at all, or that they were identical to the pancreatic cells? So, which one of these was actually discovered about nerve cells in case of teleost? Right? So what Schaller actually discovered was that in these teleost fish, there was some nerve cells which had glandular appearance, right? So glands were already known at that time. And these particular cells also looked like how glands generally do. Right, so this is a representation. You don't have to go towards the context of the representation, right? So we are not just discussing about two particular hormones, because there are other hormones also that are secreted by this gland within the brain, or we also sometimes refer to as brain itself as the gland, right? So over here, what you can see is this is the brain of the teleost fish, while this is the brain of mammals, even in humans, right? This part particularly is glandular and it secretes hormones. It is the hypothalamus and pituitary over here. The hypothalamus and pituitary have also been labeled over here. And you can see how it resembles maybe a pancreatic cells that secrete the hormone or gastric cells that secrete the hormone, right? So it is very glandular in structure. So that is what was identified in the teleost fish. So who was known as the father of neuroendocrinology, right? Was it David Rioch or Rena Popa or George Wislocky or Jeffrey Harris? So this is more a memory-based question. The correct answer is Jeffrey Harris. This is the picture of Jeffrey Harris. He's best known for showing that the anterior pituitary is regulated by hypothalamus via the hypophyseal portal system. So we have studied this adenohypophyseal system and hypophyseal system by itself also. His work established the principles for the 1977 Nobel Prize winning discovery of hypothalamic hormones by Shally and Gilman, right? Thereafter, which term describes, one moment. Yeah. Thereafter, which term describes the process where both pre and postsynaptic activities increase in correlation, leading to learning, right? Is it Hebbian synapse or reverberatory activity or long-term potentiation or cell assembly, right? So all of these were actually postulates that David Hebb, Daniel Hebb, I don't remember the first name, had basically proposed, right? So which one of these actually goes with what the statement is asking, right? So, okay, just a moment. I'll just reshare my screen. Yeah. So the correct answer over here is Hebbian synapse, right? So what is Hebbian synapse? It is basically that once a particular series of synapse is getting activated by a particular stimuli, they form a correlation, and whenever that particular intensity of stimuli is coming, that correlation is becoming stronger and stronger, and that is how this correlation is actually getting remembered. So there is a memory of that correlation that is there. So whenever again these neurons synapse together, right, we will know that, oh, this is because of that stimulus. So that memory can be recalled, right? So over here is a representation from the book Candle by Candle, right? So in stage A, the synaptic connections between five neurons are initially very weak, one, two, three, four, and five, right? Or they are even non-existent. And here are not drawn at all. So connections over here are not shown at all. Neuron 1, 3, and 5 are about to be activated by an external stimuli. Right, so in B, an external stimuli is received, and now 1, 3, and 5 are activated. Right, so the input current activates three neurons, and Hebbian plasticity causes the synaptic connections between the neurons to strengthen and permanent associative memory storage, right? So some input is coming, it is firing, it is getting these three neurons fired particularly, and this plasticity of synaptic plasticity is remembering that, okay, these particular inputs are firing these particular neurons, right? Thereafter, what happens is when the input current ceases, neuronal activity also ceases, right? The Hebbian strengthening of connections occurs during this interval after some delay. So what happens after some delay? Input current stimulates just one of the original three neurons, right? So for instance, only one is getting stimulated. But uh, excitatory connections complete the entire pattern. So only, okay, over here only the fifth one is getting activated, but the fifth one has a very strong correlation now with 1 and 3. So it automatically activating this 1 and 3 also. All three neurons of the pattern become activated, right? After this, even after the input current over here has ended, right? The neurons remain persistently active. A non-selective inhibitory input to all neurons quenches the persistent activity pattern. So if any of them is stopped, then the entire activity pattern is stopped. Circuit returns to a quiescent state. That is a normal unactivated state, right? Then question number 8 was what part of the brain was considered the center for understanding emotions, right? So the question you have to read properly. It is not uh, where the emotions are present or which part is responsible for emotions. It is which part of the brain is responsible for understanding the emotions, right? So the cognition or where the it uh, higher cognitive abilities are at play, wherein they are understanding whatever input is given to them, right? So, is it in the hypothalamus or thalamus or cingulate gyrus or the mammillary bodies, right? So, the correct answer for this one is cingulate gyrus. So, this region is the cingulate gyrus, right? It is just above this corpus callosum, as you can see. Yeah. Now, this is a very interesting topic. So we'll discuss it at length again in the next week also, but I have kept the discussion over here as well. So what type of memory problem did HM develop after his surgery, right? Was it short-term memory loss or long-term memory loss or anterograde amnesia or retrograde amnesia? Right? So, what was the problem that he developed? So, it was actually anterograde amnesia, right? So, let us understand or discuss in detail about this particular patient. So, after HM died on December 2, 2008, his full name was actually revealed. It was Henry Molaison, and HM was a 27-year-old man and had suffered for more than 10 years from untreatable temporal lobe epilepsy caused by brain damage sustained at age 7 in a bicycle accident. As an adult, his seizures rendered him unable to work or lead a normal life, right? So, as uh, is the case in many of these epileptic patients, right? So, some of them get mild seizures, which does not really interfere that much with their day-to-day life, but sometimes the seizures are very frequent and they are, very spasmic, and it might really hinder your day-to-day life, and it might render you bedridden also at times. So that was the case for this particular patient, HM, right? So in that case, what is generally done is lobotomy, right? In which the if it is a focal seizure, that is, it is originating particularly at one point in the brain, that particular region is identified, and if it is not in a life threatening region, that if we remove that region, say your heartbeat cannot be controlled or your respiratory pattern will not be controlled, right? Which are things that is very, very crucial, right? So if it is not in any of that region, what is done is the region is removed, right? So if it is not particularly life sustaining region, it is that a small section is identified which is creating that seizure, and it is lobotomized, so it is removed, right? In case of this particular patient, what was done was at surgery, Scoville removed the hippocampal formation, which was the amygdala and parts of multimodal association area from the temporal cortex, bilaterally amygdala and parts of the multimodal. Okay, I have written it twice, sorry. Right, so this was what was removed. Over here, you can see This A is a representation of the key components of the medial temporal lobe, which are important for the memory storage. These are the perirhinal cortex, entorhinal cortex, and parahippocampal cortex, right? So these are the various regions, right? And over here are listed the particular areas that had been incised. So 1, 2, and 3 over here, as you can see, this was the lesion. So in this area, amygdala, entorhinal cortex were the ones that were, regions in this part of the brain that were removed. So the areas of temporal lobe resected, which was this gray shading in the patient known as HM, viewed from the ventral surface of the brain, uh, it looks like this. Surgery was a bilateral, single stage procedure, but to illustrate the structures that were removed, the right side of the image is shown here intact. So both these portions were actually removed, but in order to tell what were these areas, one of them is labeled over here, right? So actually both the places this particular region was removed. The longitudinal extent of lesion is shown in the ventral view of the brain. Over here, cross sections 1 to 3. Actually, this is the ventral view. Cross sections 1 to 3 show the estimated extent of the areas of the brain removed from patient HM, right? Thereafter, over here, you can see this is a CT or an MRI. So yes, it is an MRI scan of the parasagittal section from the left side of patient HM's brain. Now, this asterisk mark is the region that has been removed, and this arrow marked is a region which also performs the is related to this function that is actually left behind, right? So this much area has been removed. So calibration bar at okay, we'll see. Asterisk indicates the resected portion of the anterior lobe. The arrowhead point to remaining portion of the intraventricular portion of the hippocampal formation, and approximately 2 centimeter of preserved hippocampus campal formation is visible bilaterally. Note also the substantial uh, degeneration in enlarged foliar spaces of the cerebellum, right? Other degenerative things also you can see in this MRI. So after this was done, the seizures were better controlled, but he was left with a devastating memory deficit or amnesia, right? So what happened? So amnesia was actually better understood after this case report came into light, right? So in this patient's case, he still had normal working memory for seconds or minutes, indicating that the medial temporal lobe is not necessary for transient memory. So that short-term memory, or not short, transient memory he had. He also had long-term memory for events that had occurred before the operation, right? So he remembered his name, the job he had held, childhood events, although his memory of information acquired in years just before the surgery was not that robust. In addition, he attained a command of language, retained a command of language, including his vocabulary, indicating that semantic memory was also preserved. His IQ remained unchanged in the range of bright or to normal, right? So that was also completely there, right? So what was actually missing? What HM now lacked, and lacked dramatically, was the ability to transfer new information from working memory into long-term memory. So that potentiation, as we call it, long-term memory potentiation was not taking place, right? So he was unable to retain for lengthy periods information about people, places, or objects that he had just encountered. He asked to remember, when asked to remember a new telephone, he could repeat it immediately for seconds to minutes because of his intact working memory. However, when he was distracted, even briefly, he forgot the number, and he could not recognize people after he met after the surgery, even when he met them again and again, right? So that short term memory, working memory was there, but it was not getting locked into his brain as a memory that is retained or a long-term memory, right? For several years thereafter, he saw Milner, that is Brenda Milner, every month, right? She was the psychologist for this person. Yet each time she entered the room, he reacted as though he had never seen her before. Again, we have just discussed who was the psychologist who studied the patient HM. Was it Donald Hebb or Brenda Milner or Oliver Zangwill or Peter Milner? Right? So I have already given away the answer. It was Brenda Milner, right? She is a British, I think she's Britain born only. She's a British psychologist. Currently, she's around 107 years old. Um, yeah, and she has shown, thrown light upon the subject through the discussion of this particular patient HM. Okay, so that was the end of the first part of the session. We can move on to the discussion portion. Yeah. So in the discussion portion, as I briefly introduced in the first half, what we'll be doing is we'll go forward and we'll understand the sensory system. We'll talk about the various senses, how those senses are actually perceived by our brain, where in our brain they are perceived, most of which the where part we have actually already discussed in the previous session. The how it is perceived is what is going to be the question over here. So what is being perceived and how it is being perceived. Right, that is what we are going to talk about our five major senses. Vision, auditory, olfaction, touch, and gustation, right? Gustatory senses. So these are the ones that we are going to talk about. It will take some time, so we'll begin with it. Just give me a short moment. I'll just return, um, and we can begin this. Reshare the screen. Yes. So we'll just begin with this portion, right? So this is the part that we have already discussed. I've just kept it so that you can recall that in the previous section, the first, I think, 4 or 5 slides we have already discussed. We have talked about different kinds of receptors, chemoreceptors in which uh, sorry, the stimulant is some kind of a chemical, photoreceptors wherein photons are the stimulants, and mechanoreceptors where some kind of a pressure could be the stimulating factor, right? We have also briefly discussed about the receptive fields of these neurons, and this chart also we had discussed, right? So now we'll begin with vision, right? We'll understand what is vision, how it is perceived by our brain, right? And we'll actually, in the case of vision, we'll see some very interesting portions also, right? So vision, in case of this, although eyes play an undeniably crucial role in vision, the brain is also involved in active actively constructing a visual representation of the world that is not a literal reproduction of the pattern of light falling on the eyes, right? So you can imagine, when I ask you to imagine something, I am not showing you a picture. I am simply, knitting a picture that your brain is actually forming for you, right? So if I say, imagine that I was sitting with a beautiful sunset in my background. And it would be of a good orange color. And in these okay, my brain is also very exhausted. I can't draw. I can't uh, make you imagine a picture right now, right? So that is the extent of the imagination. So I am sitting over here, and my background is a beautiful sunset, right? So if you have to imagine that, some might imagine the sunset as being that pink, violet color, as we sometimes see the sky is, right? Some might see it as bright red, some might see it as yellow. So that is the different representations of visions that your brain is bringing to life, right? So sometimes the brain is also heavily involved in this process. It is not just the eyes that are playing that role, right? So the human perception of colors result from the simultaneous activation of three different classes of photoreceptors in the retina. So we have three different classes of photoreceptors, and apart from that, we have another photoreceptor which is rods, right? So there are three types of cones that we have and one rod that we have, right? So over here you can see that the three types of cones are the blue, green, and red cone, right? Which is why most of the colored images also that you will see, or earlier they were known as RGB images only, right? Even if you do microscopy to this day, those images or those channels are known as RGB only. So you can see like a plethora of colors, but the major, primary cones or primary colors that are visible to us should be red, green, and blue. The combination of these help form different colors, right, from their simultaneous activation. So the blue has a peak of at around 437, green has a peak at around 533, and red has a peak at around 564, right? And you can see green and red are overlapping more than blue per se. And rod, which is mostly active at night, you can see it has a wider range as compared to any of the cones, right? Because it should be able to, because it is like one single kind of photoreceptor will respond to that dim light, so it should be able to respond to a plethora of ranges. So that is why it has a wider range of coverage or receptive field, right? This is another representation how the primary cones are green, blue, and red, and when they are activated at different levels, they produce different colors such as yellow, pink, white, etc. Right? Then there are spatial properties of scenes and objects which are conveyed by populations of neurons, each of which represent a small component of the image itself. So you can see that all these three represent the same image, right? When we have limited number of boxes that are fitted into the entire image, you can see it as a very pixelated or unclear kind of an image. When the number of boxes that are fitted to this image are increased from the previous one, you can see it becomes a little more clear, but it still appears what we call pixelated, right? But when we substantially increase the number of boxes that represent a single, portion in this picture, you can see how clear it has become. Although all these three are the representation of the same region, right? Now, what has changed from figure A to figure C? Over here, what has happened is that the box size has reduced because the same amount of area is covered by more number of boxes than less number of boxes over here. So over here there were like 400 boxes in this entire region. So one box had to cover a larger area, right? Over here there are like 16 and four zeros. So 1,60,000, if I am correct, or 16 lakh boxes in this entire image, right? So each box is actually covering a very small area. That is why it is very clear, right? Thereafter, we come to the discussion of sensation versus perception. Right, so sensation actually refers to effects of stimulus on the sensory organs, whereas perception involves the elaboration, interpretation of that sensory signal based on, for example, knowledge of how objects are structured. Right, so a common example that I would like to give you is um, if I show you, for instance, my earphones, right? So you might arrive to the conclusion that this is a earphone, or at least you will say that it is a gadget, or you might say that it is something that is squarish looking and maybe metallic looking, right? So, but what am I showing you? I'm just showing you the structure, right? If I show the same structure to say a 3-year-old who has never seen any such kind of gadget, they might not think in the same way that you are thinking, right? They might not know that it is an earphone. They might not know that it is something, to work with or to play with. They might pick it up and try to eat it also, right? Because they don't have that understanding or previous knowledge which tells them that, okay, this is a gadget, not something to eat or not food itself, right? So, but the sensation in both cases was same, right? The same stimulus was actually shown to the person, but how it was perceived was different based upon prior knowledge that the person might have had, right? So, this, for instance, right, is a very good example. Over here, what is actually drawn is, some part of a circle, another, circle like that, another circle like that, and three, brackets, right? But that is what is actually drawn over here. But when you look at it, what does it appear like? It appears as if there were three circles over here and there was one triangle over here with this line, it was shown. And on top of that, there is an opaque white triangle that is lying, right? So that is what we perceive when we first look at this image. What is actually shown is, or what how it is actually made is through these different brackets put together, right? And through these different circles put together, right? Half the circles or some portions of the circles put together. It is not actually, those circles and then a triangle and then an inverted triangle on top of that. That is not what it is, right? So do you automatically perceive a white triangle that really isn't there, right? So this is called the Kanis Kaniza illusion, right? So this is one of the illusions. Similarly, this is another kind of an illusion, right? Over here, you can see how our brain perceives it is there were multiple circles that were present, and on top of that, a cuboid kind of a structure is drawn, right? But that is not actually the case, right? Or it might not be the case, or it may be the case. We don't know, right? Then this another is a very good example from Professor Nitin Gupta's course, which really brought light to this concept for me, right? So try to make sense of what this is. Can can anybody tell me what this image is? If anybody had to guess. I'll anyway tell you. Okay. So if there are no cases, I would first like to give certain hints and then we'll come to the conclusion of what this is. Right? I can't hear you, Ranjini. I just saw, I think you are muted. Still muted. Let me just unmute you.
Pieces, broken pieces.
Broken pieces, right? That is what it looks like, right? But you will see the beauty of our brain once I reveal like another part wherein these pieces are actually kept intact, but something is overlain on top of it, right? Similar to the illusions that we were seeing previously, right? So, for instance, it has been changed to this, wherein all of these pieces are still intact but something has been overlain on that image. Now, what does our brain perceive it to be, right? It probably perceives it to be the letter B, right? Which was not apparent in this first image, but in the second image, because our brain automatically is thinking that there is something on top of it because of which those pieces are getting hidden, right? But it seems like that pattern that they were forming was this letter B, right? Although there is no additional information apart from this black ink like overlay, other than that, there is no additional information between the first image and the second image in that yellow portion, right? So you can even match it. There is no additional image. But see how our brain perception has suddenly changed, right? So that is the beauty of perception. And this is a classic, very good example of how sensation is different from perception, right? So let us also understand the structure of the neural cells in the eye, right? So the eye, as you know it, looks something like this. We'll not go into the detail of exactly understanding the anatomy of the eye over here. We'll revise quickly that there are three major layers. The sclera is one of them, retina is one of them, and the conjunctiva is one of them, right? So, the neural cells in the eye, they lie in this layer, the retina, right? You can see this retina is actually leading eventually to the optic nerve, which is going to take the signal to the brain, right? So, the retinal layer also has multiple neural cells. So, retina is the internal surface of the eye that contains specialized photoreceptors that convert light into neural signals, right? The photoreceptors are made up of rod cells, which are specialized for low level of light intensity, such as those found at night, and the cone cells, which are more active during daytime and are specialized for detecting different wavelengths of light, from which the brain can compute color, right? As we discussed when we were looking at the ranges. So if we look at this eye, this retinal layer particularly, right, what we can see is, okay, sclera and choroid. Choroid was the one that I wanted to see, not conjunctiva. Conjunctiva is that the uh, transparent portion in front of our lens, not that. Sclera, choroid, and retina are the three layers. So this one is sclera, this is choroid, and this entire one is a retina, right? So retina has been expanded over here to tell you what are the specific cells that are present over here. So there are retinal pigment epithelium cells, there are rods and cones, there are horizontal cells, which are basically uh, helping in connections between these rods and cones and also between these bipolar cells. Then there are the amacrine cells and the ganglion cells, which further leave the nerve fibers of these ganglion cells basically go and form the optic nerve, right? So there is already a stage of neural computation that takes place at the retina itself. Now, what is this, right? So as you can see, there are multiple kinds of these horizontal cells, also ganglion cells, horizontal cells, right? We'll come to that. So bipolar cells over here, which are transferring information from the rods and cones to these ganglion cells. Uh, they are in the retina are a type of neuron that behave in one of two ways. They either detect light areas on dark backgrounds, which is on situation, or they detect dark areas on light backgrounds, which is off situation, right? For instance, you can consider this slide to be the light area, and this is a light background, and this is the dark area on that light background. Or on the contrary, if the slide background had been black and I would have probably written this with white, that would be the white area on the dark background, right? So higher level of processing by retinal ganglion cells has more complex set of on and off properties. Most retinal ganglion neurons have a particular characteristic response to light that is termed as center surround receptor field. So this is where receptive field of the retina comes into picture, right? So the term receptor field denotes the region of space that elicits a response from a given neuron, right? One intriguing feature of the receptor field of these cells and many others in the visual system is that they do not respond to light as such. They respond to differences in light across the receptor field, right? So if there was just light that was coming in, that is not what these neurons will respond to. They will respond to the differences in light, right? So, light falling in the center of the receptor field may excite the neuron, whereas light in the surrounding area may switch it off. But when light is removed from this region, the cell is excited again, right? So, this is when the center light is falling and surrounding light is not falling. So, on center and off surround condition, right? So, in the middle, it is on because light is falling. In the surrounding it is off, right? Currently when the light is actually falling, right? The other, the reversal of this is the off center on surround, right? Other retinal ganglion cells have the opposite profile than what we studied, the on center off surround cells. Light over the entire receptive field may elicit no net effect because the center and surround inhibit each other, right? So that should also not happen. So these center surround cells form the building blocks for more advanced processing by the brain, enabling detection of, among other things, edges and orientation. So that is a feature of these only, right? There is also a blind spot in the retina, as you may be aware of. The output of the retinal ganglion cells is relayed to brain via the optic nerve. The point at which the optic nerve leaves the eye is called the blind spot because there are no rods or cones in this area, right? Okay, now this is an experiment that you can do later, right? It is an experiment to find your blind spot, right? So what you have to do is, there will obviously be a recording of the session also, or you can take a screenshot of this particular, slide. And what you can try later is that currently from the distance you are viewing your slides, I assume that you can see both a plus sign and a small circle on two ends, right? Now, what the activity is that you have to look at the plus sign only. In your peripheral vision, you will still be able to see the circle, right? But if you lift your laptop or your tablet or your phone, whatever you are viewing this on, and you try to reduce the distance of viewing, right? There will become a point at which when you are focusing your, attention to the plus sign, you will no longer be able to see the circle in your peripheral vision. That is where the blind spot is, right? So the circle that will be falling on your the place where your blind spot is, that light will be falling there, which is why be unable to see that circle in that particular frame, right? So you have to keep bringing it closer to you until that happens. So that is how you determine that this is a blind spot. Okay, then we come to phobia. It is almost opposite of the blind spot. It has a highest concentration of cones at this point, and the level of detail can be perceived is the greatest at this point. The rods are more evenly distributed across the retina, but are not present at the fovea region. Then we come to the primary visual cortex and the geniculostriate pathway, right? The dominant visual pathway in the human. Uh, just a moment. Yeah. The dominant visual pathway in the human brain travels to the primary visual cortex at the back or posterior of the brain via a processing station which is called the lateral geniculate nucleus, right? LGN. So this is the lateral geniculate nuclei on both sides of the brain. The neural representation in the LGN divides up information on the retinal surface in a number of interesting ways, right? You can see that some information from this eye is coming to this part of the brain, some is going to the other part of the brain. So it is dividing it in a specific manner. Objects in the right side of the space, termed as the right visual field, fall on the left side of the retina of both eyes and project to, so this is on the right side, right? This orange color. In the retina of both the eyes, it will fall towards the left side, right? And it will eventually be taken to the left side of the brain, right? And it will project to the left lateral geniculate nucleus, right? Similarly, this yellow one, which is on the left side, falls on the right side of the retina of both these eyes, and it is taken to the right side of this lateral geniculate nucleus. So this information is segregated into 6 different neuronal layers of the structure, 3 for each of the eye, right? The layers of lateral geniculate nuclei are not only divided according to the eye, but also contain a further subdivision, right? The upper 4 layers have small cell bodies and have been termed as parvocellular or P layers, whereas lower 2 layers contain larger cell bodies and have been termed as magnocellular or M layers, right? So these parvocellular cells respond to detail and are concerned with the color vision, while magnocellular are more sensitive to movement than color and respond to larger areas of visual field. So they have different specific functions, which is why they are different also. More recently, a third type of cell, which is K or coniocellular cell, has been documented in the lateral geniculate nuclei. It lies between magnocellular and parvocellular layers, and these cells show much less functional specificity than magno and parvo cells and have a different pattern of connectivity also. Right? Then we come to the discovery, the Nobel Prize discussion, or the discovery of Torsten Wiesel, who was a Swedish neuroscientist, and David Hubble, who was a Canadian neurophysiologist, right? So they shared a Nobel Prize in Medicine in 1981, and they elucidated the properties of neurons in the primary visual cortex, right? So this was their study. So let me remind you, we have already discussed it in brief in Sir's lectures also. What they did was they studied the cat, right? They gave certain stimuli. They had a recording electrode in the brain of the cat and they studied this, right? They found that particular neurons in the visual cortexes of cats and monkeys, the areas in the brain responsible for processing visual information, didn't respond to simple points of light, but rather to lines, and in particular lines and contours with specific orientations, right? So, some neurons responded to horizontal lines, others to vertical, and still others to orientations in between these two conditions, right? In addition, they found neurons responding to signals from both eyes, but usually one or the other would dominate. Over subsequent years, Hubble and Wiesel refined their understanding, mapping the visual centers of the feline and primate subjects with increasing precision. And they found that the visual cortex consisted of narrow columns of cells organized by eye preference and response to orientation, which they termed ocular dominance columns and orientation columns. They formed an elegantly organized functional map of the neurons that could process the complex input arriving from both of the animal's eyes, right? So, this was a simple cell, right? A simple cell in V1, which is this visual cortex area, primary visual cortex area in the brain, responds to lines of particular length and orientation. Its response may be derived from a combination of responses from different cells with center surround properties, such as those located in the LGN, right? So these simple cells envision the cells that are responsible to respond to light in a particular orientation. Then there are also complex cells in vision. These are cells that respond to light in a particular orientation but do not respond to single points of lights, right? And hypocomplex cells are those cells that respond to particular orientation and particular lengths. So they are very, very specific, right? Then if you see this pathway, you can see convergent connections allow the cortical neurons to abstract complex information from simple patterns, right? So this, for instance, is the input, right? And the output is so this particular cell in V4 is getting input from this neuron and this neuron, right? This is giving it an input of a horizontal line and this is giving it an input of a slanting line, right? This one is getting input from this and this, which is giving it an input of a V-like structure, right? This one is getting this kind of an input, one from this neuron, one from this neuron. And all of them say, even if they are getting inputs from two different structures, the overall input can lead to the output that is actually two different kinds of triangles, right? So I hope you understand that there are simple connections, but if they converge their information to the higher processing regions, this convergent information is what lets us make sense of what is happening in front of us, right? Then we come to another sense altogether, which is sound. So we did spend a lot of time in vision itself. Let's go through sound. Sound originates from the motion or vibration of an object. For example, the vibration of the vocal cords, the plucking of a violin string, or the phasing of an overhead aircraft, passing of an overhead aircraft, right? The human auditory system is capable of detecting a huge range of changes in the air pressure from around point 0.00002 to more than 100 pascals. And however, the role of the hearing brain is not merely to detect such changes, right? As with vision and other perceptual systems, goal of hearing is not to create a literal depiction of the outside world, but rather to construct an internal model of the world that can be interpreted and acted upon, right? So if I'm saying something, some frequency waves are leaving from, my vocal cords and they are reaching your ears and they are entering your ear. That frequency is not what is being read, right? Your brain is making sense of what that frequency means altogether, right? So that is why it can hear words instead of just frequency of a particular frequency of sound, right? This model is constructed not only for the ongoing sensory information, but also from previous sensory experiences, right? So, I might be speaking in English, but if you don't know English, right, it might be sounding like I'm blabbering to you, right? For instance, you know, I might be a native Tamil speaker and none of you know Tamil, right? So if I speak in that language, it might sound like blabbering to you because you will not understand what I'm saying, right? So for you to understand what I'm saying, you should know the language. That is your previous experience, right? For example, we recognize a familiar tune when presented in a different key, and we can recognize a familiar voice in a wide range of acoustic environments also, right? So, even in our noisy environments, our ears often encounter several sounds at once. But it is the job of the brain and not the ears to figure out how many of these different sound sources there are and what they correspond to. This depends both on the incoming sensory information and learned knowledge about the sounds. And some might be filtered out also. The function of this action is performed by what? Reticular activating system, the ascending portion, if I'm not wrong, right? Then, when we talk about perception, if one is listening to a very familiar song, such as Waving Flag by Khan, right? But there are gaps of 2 to 5 seconds in that song, which generally, if you go to concerts, these singers do, right? So, suppose somebody is in that tune and singing When I Get Older, and the person stops, right? If you're familiar with that song, there are more active areas in your brain which will actively work to fill in that gap, right? You might, sing it out loud to fill it, uh, or you might constantly be thinking in order to recall that information, that what is the next word, what is the next line, right? So there will be more active areas in your brain as compared to if it was an unfamiliar tune or unfamiliar song, right? So that is the difference between the sensation and perception over here, right? So our musical and lyrical knowledge can also fill in silent gaps in heard songs, right? One difference that does exist between this auditory and visual senses is their sensitivity to temporal and spatial information, right? So, for instance, auditory system is exquisitely tuned to detect temporal information, that is with time information that is changing, such as rapid changes in frequency that characterize certain speech sounds, sorry, and in grouping information together over time, such as in extracting melody from the music, right? In different time intervals associated with dots and dashes in Morse code are much easier to process when heard than seen, right? So Morse code can be heard also and it can be seen also, and it has been found that it is easier to process when it is heard rather than when it is seen. So temporal information is probably better tuned by auditory system, right? In contrast, it is generally much easier to locate an object in space with vision than with hearing, right? So if I close my eyes, I try to hear that, oh, I hear a familiar sound. Okay, I'll be drawn towards that sound rather than just opening my eyes and see where the that particular noise or sound is coming from, right? So spatial information wise, our this thing vision is better tuned, right? What is the nature of sound? One of the simplest sounds has a sinusoid waveform. When pressure change is plotted against time, it gives a sinusoid waveform, and these sounds are termed as pure tones, right? Pure tones have a characteristic pitch that is related to frequency of the sound wave, and the human auditory system responds to sound frequencies between 20 hertz to 20,000 hertz, right? Below this, it is known as infrasonic for us, and above this, it is ultrasonic for us. The intensity of sound, that is its amplitude, when considered as a sine wave, is related to subjective experience of loudness, right? For me, for instance, 50% sound in my earphone might be very loud, and for some other person, 50% might be too low, and they might be hearing in the same model of earphones, they might be more used to maybe 80% sound, right? So for them, 50% will be low. So it is relative. So this is a pure tone. I'll just skip this portion, right? Yeah. Then we come to perception. In perception, it is crucial to make a distinction between physical properties of a stimulus and its perceived characteristics, as we saw in case of vision also. In vision, there is a close relationship between wavelength of light, which is physical property, and color, which is a psychological property, right? So something that we perceive as pink is not actually pink. It is just emitting that wavelength of light. And our eyes looking is determining that, oh, this is pink. But the two things are not the same, right? So it is possible to see color without its associated wavelength, as in after images, and it is possible to process wavelength without perceiving the color, as in cerebral achromatopsia, right? So these are two things that you can study more about. When we come to sound perception in hearing, although pitch is related to frequency of sounds and loudness, it is related to intensity or amplitude of sounds, right? Pitch and loudness are regarded as psychological features of sound, whereas frequency and intensity are the physical properties. Although amplitude and frequency might be independent physical properties of sound waves, but the subjective properties most closely associated with them, that is pitch and loudness, are not processed by the brain in a completely independent way, right? In everyday life, pure tones are seldom heard, right? However, many sounds can be described in terms of combinations of superimposed sinusoids of different frequencies, intensities, and phases. For example, musical notes typically contain a series of regularly spaced sinusoids, right? The lowest component is termed as the fundamental frequency or F naught, and it typically determines the perceived pitch of a musical note. So, this is what it means. The pure tone, which is over here, is consists of sinusoidal varying pressures, right? Many naturally occurring sounds, such as musical tones in the bottom, consist of regular series of sinusoids of different frequencies, and the perceived pitch is related to the lowest frequency in the series, right? This is F naught. The relative intensity levels of different sinusoid components of musical sounds are important for discriminating between the same notes played on different instruments, right? Then let us actually come to how this information is transferred from our ear to our brain, right? So, overall, we might remember that the ear contains three main parts: the outer ear, middle ear, and inner ear. The outer ear has this pinna, the ear which consists of the ear free earlobe, also the auditory canal. The middle ear has these three bones, malleus incus or hammer, anvil, stirrup, right? And uh, the inner ear has the semicircular canals and the cochlea, right? So this is what we have already talked about. Outer ear is responsible for reflections of sound waves within the folds of pinna, and with auditory canal can amplify certain sounds and are important for locating a sound source. Middle ear, these bones that are present transfer the mechanical pressure on the eardrum at the end of the airborne auditory canal to a smaller membrane called the oval window over here in the fluid-filled cochlea. Right? The inner ear contains chambers that are important both for senses of hearing, that is the cochlea, and balance, which is the semicircular canals. The cochlea converts liquid-borne sounds into neural impulses. The membrane within the cochlea is termed as a basilar membrane, which contains tiny hair cells linked to the receptors. The sound induces mechanical movement of the basilar membrane and the hair cells on it. These movements induce a flow of ions through stress-sensitive ion channels that initiate neural activity or release of neurotransmitters. The basilar membrane is not uniform but has different mechanical properties at either end. And the end nearest to the oval window is narrower and stiffer and shows a maximal deflection to high frequency sounds. The end nearest the center of its spiral shape is wider and more elastic and shows a maximal deflection to low frequency sounds. As such, different parts of membrane are sensitive to different frequencies of sounds, and sounds originating from different parts of space do not stimulate different parts of the membrane, right? However, location of sound sources needs to be inferred from other kinds of information, that is differences between signals in the ear, right? So for instance, I will know that the sound is coming from my right if there is more stimulation in my right side of the ear than my left side of the ear, right? So that is how the distinction is generally made. There are 4 or 5 synapses in the auditory pathway from the ear to the brain, right? Over here, you can see there's the auditory nerve. There's this first synapse, the ventral cochlear nucleus, and the dorsal cochlear nucleus when it is transferring this information to. Then comes the superior olivary nucleus, then the inferior colliculus, then the medial geniculate nucleus. The lateral geniculate was for the eye, and medial geniculate is for the auditory one, and this is taking it to the auditory cortex of the brain, right? This auditory cortex is known as A1 or the core region, as opposed to the V1 or primary visual cortex in case of the um, vision, right? The primary auditory cortex is located in Heschl's gyrus in temporal lobes and is surrounded by adjacent secondary auditory cortical areas called the belt and the parabelt regions. Right? These secondary regions also receive some input from the medial geniculate nucleus, and hence damage to the primary auditory cortex does not produce complete deafness but leads to problems in identifying and locating the sounds. Right? So this is A1, the primary auditory cortex, and A2, right? Secondary auditory cortex, which is belt areas. This is parabelt and this is belt. So in the parabelt, you can see there are different frequencies. So these are neurons that recognize these frequencies of sounds, right? So while cochlear nucleus has 90,000 neurons, while the medial geniculate nuclei has 50,000, and auditory cortex has over 100 million, right? The ascending auditory pathway is not a passive transmission of the information of the ear, but rather involved in active extraction and synthesis of information. And that is what the brain does with all the signals that it is getting. In addition, there are descending top-down pathways that go as far back as cochlea itself and may be important in auditory attention. Right? Over here, uh, as we were mentioning, this is the basilar membrane, right? So there are different frequencies of sound which are understood by different kinds of um, okay, this basilar membrane is within the cochlea. So it's generally like this. If we unroll it, we can see it like this, right? Early auditory system can be said to have a tonotopic organization, right? Because different frequencies at different levels it is getting. Just as different parts of the basilar membrane respond maximally to different sound frequencies, neurons within the auditory nerve respond maximally to certain sound frequencies more than others. Right? The nerve bundle is orderly such that neurons responding to higher frequencies are located on the periphery and those responding to lower frequencies more centrally. So, to some extent, this organization is carried upwards to early cortical stages, right? In both humans and other animals, there is evidence that central region of the primary auditory cortex responds to lower frequencies and outer regions on both sides to higher frequencies. So over here again, I did explain how these belt area has different frequencies at different regions. So if we compare broadly the auditory versus visual systems, the auditory system takes the medial geniculate nucleus, which projects to the primary auditory cortex, and the visual, on the other hand, takes the this root lateral geniculate nucleus, which projects to the primary visual cortex. Then, if we organize these neural processing regions, there is tonotopic organization in the auditory system, that is orderly mapping between sound frequency and the position on the cortex, while there is retinotopic organization, that is orderly mapping between position on retina and position on cortex in this case, in the visual system, right? Then again, if we talk about the temporal versus spatial sensitivity, the auditory system is more temporally sensitive than spatially sensitive, while the visual system is more spatially sensitive than temporally sensitive, right? Then the functional specialization of the feature processing, the auditory system, it is less well documented in the auditory domain, while it is well documented for color and movement in case of the visual system. If we talk about higher order context dependent pathways, then in auditory system, there is evidence for separate auditory pathways for what versus where and how. And in case of visual system, there is evidence for separate visual pathways for again what versus where and how, right? So touch is another sense that we come to now. What is touch? It is defined as a direct contact between two physical bodies. In neuroscience, touch refers to the special sense by which contact within the body is perceived consciously. It can be active as when you move your hand or some other part of your body against another surface, or passive when someone or something else touches you, right? So passive touch is used for naming objects or describing sensations. Active touch is used when the hand manipulates objects, such as me moving this mouse to particularly, move this cursor, the laser cursor. So active and passive modes of tactile stimulation excite the same population of receptors in the skin and evoke similar responses in afferent fibers. They differ somewhat in cognitive features that reflect attention and behavioral goals during the period of stimulation. So the hand has four types of mechanoreceptors. These are Meissner corpuscles, Merkel cells, Pacinian corpuscles, Ruffini endings, right? The sense of touch can be understood as combined result of information provided by these four acting in concert, right? So together they act, and that is how we feel the touch. So this is Meissner corpuscle RA1, Merkel disc receptor SA1, Pacinian corpuscle RA2, and Ruffini ending SA2, right? So each receptor corresponds in a distinctive manner depending on its morphology, innervation pattern, and depth in the skin, right? These receptors are innervated by either slow adapting or rapidly adapting axons. So slow adapting is SA and rapidly adapting is RA, right? So slow adapting or SA fibers respond to steady skin indentation with the sustained discharge, whereas rapidly adapting RA fibers stop firing as soon as the indentation is stationary, right? So sustained mechanical sensations from the hand must accordingly arise from the SA fiber, and sensation of motion across the skin is signaled by RA fibers, right? So, for instance, I know that I am wearing my watch, right? So that is a response of the SA fiber because it is a sustained stimulus that is being applied to my wrist area, and it is not going away, right? So it could be a response of the sustained fiber over here. Type 1 fibers terminate in clusters of small receptors in the superficial layers of the skin and the margin between dermis and epidermis, right? Type 2 fibers innervate the skin sparsely and terminate in single large receptors in the deeper layers of the dermis or subcutaneous tissue. RE1 fibers are the most numerous. So RE1 was what? Meissner corpuscles. Most numerous tactile afferents in the hand, reaching a density of approximately 2 per millimeter square at the fingertip in man and monkey, right? So these are the four types of mechanoreceptors. The Merkel cells uh, are located at tip of epidermis. Sweat ridges, the axon diameter is around 7 to 11. Conduction velocity is highest over here, 40 to 65. Its best stimulus is edges or points, and it has sustained response with slow adaptation, and the frequency ranges 0 to 100, and the best frequency 5. Threshold for rapid indentation is around 8, right? So similarly you can go ahead and read the other ones also. So this is type 1 fibers are SA1 RA1, type 2 are SA2 and RA2, right? So receptive fields in human hands are the smallest at the fingertips, right? So for instance, at the fingertips, I can feel very minute sensations also, but at my palm, I will not be able, like the receptive field will be larger, right? Less number of neurons will be present over here than here, right? So they are more densely packed in the fingertips. There are more nerve endings over here than they are at the palms, right? Because over here we use our fingers for fine motor activities also, right? So tactile information is processed in the central touch system. This comprises the dorsal column tracts of the spinal cord and relay nuclei in the brainstem and thalamus, and a hierarchy of regions in the cerebral cortex as well. The tactile information enters the cerebral cortex through the primary somatosensory cortex in the post-central gyrus of the parietal lobe. So this is uh, the one that is just behind that motor cortex, primary motor cortex, right? So this is the somatosensory cortex, just behind the motor cortex, as I told you. The neurons in SI are at least three synapses beyond the receptors in the skin, as opposed to five synapses beyond in the auditory system. Their inputs represent information processed in the dorsal column nuclei, the thalamus, and the cortex itself. So each cortical neuron receives input arising from receptors in a specific area of the skin, and these inputs together are its receptive field. So we perceive that at a particular location on skin is touched because a specific population of neurons in the brain is activated, right over here. This experience can be experimentally induced by electrical stimulations of the same cortical neurons, right? So it is a two-way thing, right? So if I'm touching over here, some particular neurons in my brains are getting activated. If those particular neurons are anyway getting activated, then I will feel a sensation over here, even if there is no touching involved, right? So the primary somatic sensory cortex comprises four cytoarchitectural areas. Over here, you can see 1, 2, 3A, 3B, and uh, yeah, Brodmann's areas 3A, 3B, 1 and 2, right? These areas are extensively interconnected such that processing of sensory information at this level involves both serial as well as parallel processing. Neurons in the areas 3A and 3B receive inputs from the lateral and medial zones of ventral posterior nucleus of the thalamus and project their axons to areas 1 and 2, right? So they'll take this information to area 1 and area 2. The areas 3B and 1 receive information from receptors in skin, whereas 3A and 2 receive information uh, uh, sorry, receive proprioceptive information from receptors in muscles, joints, and skin, right? So, somatosensory information is conveyed in the parallel from four areas S of somatosensory cortex to higher centers in the cortex, including secondary somatosensory cortex and the posterior parietal cortex and primary motor cortex. This is a columnar organization of the somatosensory cortex. We have studied this columnization of these senses also, right? Uh, then we come to the final senses, smell and taste. So this is even more brief than the one that we just talked about, right? So through the sense of smell and taste, we are able to perceive a staggering number of and variety of chemicals in the external world, right? These chemical senses inform us about the availability of foods and the potential pleasure or danger, right? Smell and taste also initiate physiological changes required for digestion and utilization of food. And in many animals, olfactory system also serves as important social function by detecting hormones that elicit innate behavioral or physiological responses. Right? Large number of olfactory receptor proteins initiate the sense of smell. If we talk about odorants, what are odorants? They are volatile chemicals that are perceived as odors and are detected by olfactory sensory neurons in the nose, right? The sensory neurons in the nose are specialized olfactory epithelium that lines part of the nasal cavity over here, approximately a 5 centimeter square area in humans, and are interspersed with glia-like supporting cells, right? So these are the supporting cells, these are the basal cells, these are the olfactory sensory neurons, right? And the axons are going in to the olfactory bulb. The olfactory sensory neuron is a bipolar nerve cell, right? A single dendrite extends from the apical end. So this is a dendrite, right? It extends from the apical end to the epithelial surface where it gives rise to numerous thin cilia that protrude to into the mucus that coats the nasal cavity, right? These cilia have receptors that recognize odorants as well as the transduction machinery needed to amplify the sensory signals and transform them into electrical signals in the neuron's axon, which projects from the basal pole of the neuron to the brain. So over here, these bipolar neurons, they are basically going across this cribriform plate, which is a bone over here, right? And into the olfactory bulb, which is a part of the brain itself, and they are going and into this glomeruli-like structure, right, wherein they are synapsing with the tufted cells and the mitral cells, which are further carrying this information to the olfactory cortex in the brain. The axons of olfactory sensory neurons pass through the cribriform plate, a perforated region in the skull above the nasal cavity, and the axons then synapse in the olfactory bulb, the first relay in the olfactory pathway, right? So this is the first relay. Humans have approximately 350 different odorant receptors, while mice have approximately around 1,000 odorant receptors. So, although odorant receptors belong to G protein coupled receptor superfamily, they share sequence motifs not seen in other superfamilies, right? So, they are different, a little different in their motif organization than other receptors of the GPCR family. Significantly, alternate receptors vary considerably in the amino acid sequence, right? A second smaller family of chemosensory receptors is also expressed in the olfactory epithelium itself. These are called trace amine associated receptors or TARS and are G protein coupled, but the protein sequence is unrelated to that of odorant receptors completely, right? They are encoded by a small family of genes which is present in human, mice, as well as in fish. So, different combinations of receptors actually encode different odorants, right? So, for instance, if only this purple one is there, I might, get the feeling that the odor is rancid, sour, and goat-like. But if the purple is also combined with this orange and green, it might, smell completely different and smell like violet, sweet, and woody, right? Similarly, different combinations of these odorants give different types of odors, right? That we perceive. So odorants are encoded in the nose by dispersed neurons. Different types of odorant receptors are expressed in several core zones of the olfactory epithelium of the mouse. Each receptor is expressed in approximately 5,000 neurons that are confined to one zone. So neurons with the same receptor are randomly scattered within the zone so that neurons with different receptors are interspersed. Although one zone may have more receptor for a particular odorant compared to other zone, all zones contain a variety of receptors so that a specific odorant may be recognized by receptors in several different zones of the nose. Because each odorant is detected by an assembly of neurons that is widely dispersed across the epithelial sheath, some receptors detecting a particular odorant will be functional when part of epithelium is damaged by the respiratory infection, right? So this is an example. If the odorant receptor expresses K20, you can see that it is present on both sides, right? If it is K21, again on both sides in various places it is present. L45, these, right? A protein that actually marks all odorant receptors. OMP shows that there are odorant receptors present on both sides throughout this olfactory epithelium, right? The axons of the sensory neurons project to the ipsilateral olfactory bulb whose rostral end lies just above the olfactory epithelium. The sensory axons terminate on the dendrites of these olfactory bulb neurons within the bundles of neuropil called glomeruli that are arrayed over the bulb surface. So we just saw that earlier, right? These tufted and mitral cells were coming there. These are the glomeruli, right? Different from the glomeruli in the kidney. The axons of olfactory sensory neurons, okay, so it is the same thing is written over here. In each glomerulus over here, sensory axons make synaptic connections with three types of neurons, right? It could either be mitral and tufted relay neurons, which project axons to the olfactory cortex and periglomerular interneurons, which encircle the glomerulus, right? The axon of the olfactory sensory neurons terminates in only one glomerulus. Similarly, primary dendrite of each mitral and tufted relay neuron is confined to a single glomerulus, right? So the same mitral cell will not go to this as well as this glomeruli, right? Each glomerulus, the axons of several thousands of sensory neurons converge on the dendrites of approximately 40 to 50 relay neurons. This convergence results in approximately a hundred-fold decrease in the number of neurons transmitting olfactory signal, right? So you can see axons of neurons with same odorant receptors converge on a few glomeruli, which are, highlighted by this white expression over here and here. One odorant can activate many glomeruli. So you can see in this heat map that multiple different regions or multiple different glomeruli are getting activated by one single odorant, right? So, when we talk about odor responses in the olfactory bulb, the organization of sensory information in olfactory bulb is dramatically different from that of the epithelium itself, right? So, this was the epithelium. We saw that staining in which OMP was staining the entire, odorant receptors across the neural epithelium. So, whereas olfactory sensory neurons with the same odorant receptor are randomly scattered in one epithelial zone, their axons converge in a few glomeruli at two spots, one on either side of the olfactory bulb, as you can see over here, right? Each glomerulus and each mitral and tufted relay neuron connected to it receives input from just one type of odorant receptor, right? The result is a precise arrangement of inputs from different odorant receptors which similar between individuals. Because each odorant is recognized by a unique combination of receptor types, each odorant also activates a particular combination of glomeruli in the olfactory bulb. At the same time, just as an odorant receptor recognizes multiple odorants, a single glomerulus or a given mitral or tufted cell is activated by more than one odorant. This organization of sensory information in the olfactory bulb is likely to be advantageous in two respects. First, the fact that signals from thousands of sensory neurons with the same odorant receptor type always converge on the same few glomeruli and relay neurons in the olfactory bulb may optimize the detection of odorants present at low concentrations also. And each glomerulus is encircled by periglomerular interneurons, these ones, right? That receive excitatory input from the sensory axon and form inhibitory dendrodendritic synapses with mitral and tufted cells. Dendrites in that glomerulus, perhaps adjacent glomeruli also. Periglomerular interneurons may therefore have a role in signal modulation. Also, granule cell interneurons deep in the bulb provide negative feedback onto the mitral and tufted cells. The granule cell interneurons are excited by the basal dendrites of the mitral and tufted cells and in turn inhibit those postsynaptic relay neurons and others with which they are connected, right? So it is a feedback regulatory mechanism basically. Then when we talk about the granule cell interneurons, the lateral inhibition afforded by these connections is thought to dampen signals from glomeruli and relay neurons that respond to an odorant only weakly, thereby sharpening the contrast between the important and irrelevant sensory information before its transmission into the cortex. Right? So the olfactory bulb transmits information to the olfactory cortex. This is the way it does so, right? Finally, to the hypothalamus and orbitofrontal cortex. Then we come to taste. So taste has five submodalities or qualities. The primary function of the gustatory system is nutritional, but we don't merely see it as nutritional, right? Humans and other mammals can distinguish five different taste qualities, which is sweet, bitter, salty, salty, sour, and umami. The taste associated with amino acids, right? So protein-rich things probably taste umami. The taste chemicals or tastants perceived as sweet are associated with food high in caloric content, while those sensed as umami are indicative of protein. Consistent with nutritional importance of carbohydrates and proteins, both sweet and umami tastes elicit pleasurable sensations in humans and are attractants for animals, right? In contrast, bitter tastings, which are often found in poisonous plants, elicit an aversive response that is an innate in animals as well as human infants and likely prevents the injection of toxic substances, right? That's why a lot of us don't really eat bitter gourd or a few other, slightly bitter tasting vegetable when we are younger, but we develop that taste. Even this black pepper, green pepper, red pepper, also for that matter. When we are younger, we don't drink it, but as and when we age, we don't eat it when we are younger, but when we age, we probably become more used to it, right? So taste is often thought to be synonymous with flavor. However, taste refers strictly to five qualities encoded in the gustatory system, whereas flavor, with its rich and varied qualities, actually stems from a combination of inputs from the gustatory, olfactory, and somatosensory system. Sometimes even visual is involved, right? In the gustatory system, sensory signals generated in mouth are relayed to the brainstem and thalamus, to the gustatory cortex. Tastings are detected by taste receptor cells that are clustered in the taste buds. Although majority of taste buds in humans are located on the tongue, some can also be found on the palate, that is the upper and lower palate, pharynx, epiglottis, and upper third of the esophagus also. Right? So this is where they meet. Right? Taste buds on the tongue are occur in structures called papillae, of which there are three types based on morphology and location. Right? So these papillae might be circumvallate, foliate, or fungiform. Right? So taste buds on the tongue occur in structures called papillae, of which there are three types based on morphology as well as location. Right? Yeah. Each fungiform papilla contains 1 to 5 taste buds, while each circumvallate or foliate papilla contains around 100. Right? The taste bud is a garlic-shaped structure, this one, and it is embedded in the epithelium. A small opening at the epithelium surface, the taste pore, is the point of contact with the taste buds. Right? So this is a TEM image of the same electron microscope image, basically. Each taste bud contains approximately 100 taste receptors or taste cells, elongated cells that stretch from taste pore to the basal area of the bud. The taste bud also contains other elongated cells that are thought to serve as supporting function, as well as small number of round cells at the base, which are thought to serve as the stem cells. The taste cells are short-lived and appear to be continually replaced from the stem cell population. Although taste cells are non-neural, their contact with gustatory sensory neurons have morphological characteristics of chemical synapses, including clustered presynaptic vesicles also, right? This one. These cells also resemble neurons in that they are electrically excitable and they have voltage gated sodium, potassium, and calcium channels and are capable of generating action potentials also. So each taste is detected by a distinct sensory transduction mechanism and distinct population of taste cells. So each of the five taste qualities involves a different sensory transduction mechanism in the microvilli of taste cells. There are, however, two general types. Bitter, sweet, and umami tastings interact with GPCRs, whereas salty and sour tastings appear to involve specific ion channels. So these are ion channels is for salty and sour, and for the others, bitter, sweet, and umami, there are GPCR-based signaling. So these are different receptors, and how combination of different receptors, give different kinds of tastes. That is shown over here, right? Sensory neurons carry taste information from the taste buds to the brain. Each taste cell is innervated at its base by the peripheral branches of the axons of the primary sensory neurons. Each sensory fiber branches many times, innervating several taste cells within the numerous taste buds. Sorry. The release of neurotransmitter from taste cells onto the sensory fibers induces action potentials in the fibers and the transmission of signals to the sensory cell body. These sensory neurons carry taste information from the taste buds to the brain. The cell bodies of gustatory sensory neurons line the geniculate, petrocell, and nodose ganglia. The peripheral branches of the gustatory sensory neurons in these three ganglia travel in cranial nerves 7, 9, and 10. The central branches of axons of the gustatory sensory neurons enter the medulla where they terminate on neurons in the gustatory area of the nucleus of the solitary tract. In most of the mammals, the neurons in the nucleus transmit signals to parabrachial nucleus of the pons over here. Uh, which in turn sends signal to gustatory information to ventroposterior medial nucleus of the thalamus. In primates, however, these neurons transmit gustatory information directly to the taste area of the thalamus. From the thalamus, taste information is transmitted to gustatory cortex. Over here, a region in the cerebral cortex located along the uh, border between the anterior insula and the frontal operculum. The gustatory cortex is believed to mediate the conscious perception and discrimination of taste stimuli. The taste areas of thalamus also transmit information both directly and indirectly to the hypothalamus, a structure that controls feeding behavior as well as autonomic responses. So that is all. I know I rushed with the olfaction and the taste. Um, but I really wanted to cover it up so that we can move on to learning and memory in the subsequent lecture, right? Uh, so that will be all for the class from my end today. If there are any doubts, you may please ask. Um, yeah. Yes, so as I said, that will be all. So thank you for attending the class. I'll stop the recording and a very good night to everybody.